Hi, this is Group 5's presentation of DNA replication. Group members are Ryan Walt, Bradley Ponder, Amber Weesey, and John Lawless.
Fire.
DNA polymerase III. DNA polymerase III is an enzyme that catalyzes the elongation of new DNA by the addition of nucleotides to the 3' end of an existing chain. DNA polymerase III synthesizes leading strands continuously. DNA polymerase III also starts DNA synthesis at the 3' end of primer and continues in 5 to 3 direction. DNA helicase. DNA helicase is an enzyme that untwists the double helix of DNA at replication forks. It separates the 2 strands and makes them available as template strands. Primase. Primase is an enzyme that joins RNA nucleotides to make a primer during DNA replication, and it uses the parental DNA strands as templates. Primase synthesizes the short-end RNA primer. Primer. Primer is a short stretch of RNA with a free 3' end, bound by complementary base pairing to the template strand and elongated with DNA nucleotides during DNA replication. DNA polymerase I. DNA polymerase I is an enzyme that catalyzes the elongation of new DNA by the addition of nucleotides to the 3' end of an existing chain. DNA polymerase I replaces the RNA primer with DNA nucleotides. DNA ligase. DNA ligase is a linking enzyme essential for DNA replication. DNA ligase catalyzes the covalent bonding of the 3' end of one DNA fragment to the 5' end of another DNA fragment. Single-strand binding protein. Single-strand binding protein is a protein that binds to the unpaired DNA strands during DNA replication, stabilizing them and holding them apart while they serve as templates for the synthesis of complementary strands of DNA.
We used a candy model named Red Licorice Replication because we think it will help us remember the job of each enzyme better when we can associate it with a different candy. The cells need to perform replication to create sister chromatids. Our body needs more DNA created to produce more cells.
We used an article named Loading Mechanisms of Ring Helicases at Replication Origins. The article helped us understand how helicase is used in replication. The article explains that replicative helicase are the motor engines of replisomes.